So I know that sometimes students get stressed out by talking about ATP synthase, so I wanted to start this with a video of my cat, who is hot, but she's pretty happy right now. Okay, so in the last video, we talked about those proteins in the electron transport chain which pass electrons between them and some of which pump protons. So that proton gradient is on one side of the membrane, and that is the intermembrane space, and then we also have the mitochondrial matrix. And so let's take a quick look at the mitochondria as a whole. So this is the mitochondria. I'm sure everyone has seen an image of this before. It kind of looks like a bacterium. It probably used to be a bacterium someday way back in evolutionary history. We see that it has an outer membrane and an inner membrane, and inside of the inner membrane is the matrix, and then the intermembrane space is this area here. So that is just to orient us where we are in the mitochondria. And so we said that the pumping of the protons occurs and increases the proton gradient into the intermembrane space. So the intermembrane space is high in protons, and the matrix is low in protons, as we could see from this image in the textbook, which I think is in the how do we know section in that chapter. So you don't need to read that entire thing, but if you want to take a look at this diagram, it might be helpful. And so again, we have high levels of protons in the intermembrane space, and then low number of protons in the matrix. The only way for the protons to cross the membrane along their concentration gradient is through this protein called ATP synthase. Now, let's just take a moment to remember that protons cannot cross the membrane because they are charged, although they are small. And we said that small molecules can pass through the membrane, but not charged small molecules. And these protons, therefore, will tend to flow from high to low concentration, and the only way to do this is to get through this ATP synthase molecule. So what happens is that the ATP synthase molecule will actually bind to a proton, and then it kind of spits it out the other side. So every time it does that, it is decreasing the concentration gradient, so bringing the protons from high concentration to low concentration. And as this occurs, there is a conformational change. So yeah, we've been talking about lots of those at the end of this semester. The conformational change that occurs, or part of it that occurs, is due to that proton binding the ATP synthase, and the conformational change then affects this other part of the protein, which has already bound to ADP and the inorganic phosphate. So the conformational change actually forces the inorganic phosphate and the ADP to come very close together to the point where they actually form a bond and form ATP. I want to point out here that there are two different parts to this protein. There is this part up here, which is actually embedded in the membrane and interacts with the protons, and then there's this other part at the bottom, which only interacts with the ADP, inorganic phosphate, and ATP. Let's compare this to the image in our textbooks. And we see that the parts are clearly labeled in the textbook, which is why this is a really nice image, although the image on the right, I think, shows the interactions a little bit better. I don't care which one you prefer. You can use either. The rotor is one part of the ATP synthase. And that is the part that interacts with the protons. So we see here on the left that the rotor is made up of the light blue and the purple unit. And then on the right, the rotor is the part that looks like a bunch of, I don't know, pieces of chalk. And we see then it's also composed of what is labeled the rotor shaft. So the rotor, again, is what interacts with the the protons, and the protons binding and being released causes the rotor to turn. So this is like a little machine, and basically because of the chemistry of it, when the protons are bound, then there's a conformational change that makes the rotor turn. And so the protons actually go for a little bit of a ride. This is like a proton carousel, and they are bound and then conformational change. And then a new one is bound, conformational change. A new one is bound, conformational change. And this occurs until the first proton gets all the way to almost where it was bound. And then there is a place where it can be released, a little channel where it can be released, and it's released again on the opposite side of the membrane. So again, the turning is important, first of all, because it is what actually allows the protons to get to the other side of the membrane. Second of all, the turning is important because the rotor actually interacts with the other subunit of the protein, the other part of the protein. And this is the stator. And so we see that it's this big chunky part on the bottom here on the right side, and then it's shown as the green and the yellow parts on the left side here in your textbook image. And there is a mechanical interaction between the rotor and the stator, and it's that mechanical interaction which actually causes the conformational change in the stator. And this conformational change, again, is what actually causes the ATP synthesis. So again, as the stator goes through a conformational change because of the turning of the rotor, that conformational change causes the phosphate group to be added onto ADP. And so that's it. That's how we get ATP hydrolysis. Or sorry, not hydrolysis, ATP synthesis. I said ATP hydrolysis because there is another component of this. So if there is an imbalance or an abnormality, and we have a higher proton concentration on the bottom here than on the top, which is not how it should be, then we can go through ATP hydrolysis. And this protein, the ATP synthase, can actually work in the reverse direction to use ATP to pump hydrogens or the protons back to the other side. Sorry, I keep saying hydrogens, but they're not hydrogens, they're just protons. Is what is shown here on the right side, ATP hydrolysis using ATP in order to restore the proton gradient. So we see that there are an equal number of protons on both sides of the membrane here, and the ATP is being hydrolyzed from ATP to ADP in order to then pump the hydrogens back to the other side of the membrane, so that then we end up having higher proton concentration in the intermembrane space, which is what we want ultimately. And this can occur temporarily. It can be very localized, and that's why this can happen, because then the localized kind of abnormality can be fixed just by little work by ATP synthase. But if all of your mitochondria were experiencing this imbalance, then this would be an illness and you would be in serious trouble. So to close out, I'll show a little GIF of ATP synthase working. This GIF is actually upside down, so I'm going to start with this GIF turned upside down. So what we're seeing again is the mitochondrial membrane and the divide in the concentration gradient. So we see that there's a high concentration of protons in the intermembrane space up here, and there's a lower concentration of protons here in the matrix. The protons are these little yellow dots. Maybe let's zoom in a bit. And the little yellow dots we see are binding to the ATP synthase, this part of the rotor here, and we see that they go for their little carousel ride, and then they are finally released to the other side of the membrane. And we also see that as this rotor turns, this other portion of the ATP synthase protein or enzyme binds to these blue molecules, and then we see like these little flashes of light, which are supposed to symbolize that the phosphate group is being added to the blue molecule, which is an ADP molecule, I guess, and then the pink molecules are ATP synthase, or ATP, sorry. So yeah, that's what happens. And one more time, here is the GIF as it was originally made. So this is opposite of what we saw in our diagrams in the textbook. And we see that the matrix is on the top, the intermembrane space is on the bottom. We see the proton gradient being higher on the bottom, and we see ATP being made again by the stator and the rotor continuing to rotate. And so if you were interested in seeing those labels on the GIF itself, hopefully this helps a little bit. So finally, once again, this is a different type of ATP synthesis than we saw previously in cellular respiration early on, which was in glycolysis as well as in the Krebs cycle, or TCA. This is referred to as oxidative phosphorylation. Even though the ATP synthase does not directly use oxygen, it is oxygen which is important for maintaining that proton gradient, because unless oxygen can receive that electron at the end of the transport chain, then none of the proteins are going to be able to pass on the electron to the subsequent protein complex. So essentially there's going to be a backup in the assembly line. And so again, that's why it's called oxidative phosphorylation instead of substrate level phosphorylation. And again, as we saw in that SpongeBob meme, so there we go. Again, we have much lower levels of ATP produced through substrate level phosphorylation. Actually, it's more than 2 ATP, we know, because it's 2 ATP from glycolysis, and then we have an additional GTP, 2 GTP produced through the Krebs cycle or TCA cycle, and then we have much more ATP produced through oxidative phosphorylation.